Yes, so thanks a lot, and it's my great honor now to say hello and to introduce Leonard Jason from Chicago. So we are very happy that you agreed also to give us a presentation about your long-standing expertise on the role of the Epstein-Barr virus in ME/CFS. I think you're actually one of the pioneers in analyzing EBV and ME/CFS, and Leonard Jason published numerous papers on this topic. So please, it's your stage.
Thank you very much. Can you see the slides?
Yes, we see them well.
Yes, you can. Everything
Yes.
okay?
Yeah, we see you and we see the slides.
Terrific, thank you. My talk is titled Predictors of ME/CFS Following EBV and Implications for PCS. The key question I'll be talking about today is why some people get sick with the viral infection and stay ill versus those who get sick and recover. I believe the best way to answer this type question is to examine individuals prior to them getting sick to see if they might have some characteristics that predispose them to long-term risk. For the past decade, our group, including Dr. Ben Katz at Lurie Children's Hospital, has been working to identify immune profiles and behavioral domains that existed prior to infection of EBV. Such insights can help us with understanding the predisposing factors for ME/CFS as well as COVID long haulers. Our method was from 2014 to 2018, actually prior to the pandemic. We collected data from 4,500 healthy college students at Northwestern and followed them for the development of ME/CFS. About 5% developed infectious mononucleosis, which is what we expected. Some continued of that 5% to have severe symptoms at 6-month follow-up. This was published a couple years ago in Clinical Infectious Diseases. We categorized the individuals 6 months later into those who met one case definition, the Fukuda, which is more general, polythetic. Those who met more than one case definition, Fukuda, in either the Canadian or Institute of Medicine criteria. We called them more narrow case definition or severe ME/CFS, and those who recovered. In terms of pre-illness variables, students who developed ME/CFS versus those who did not, following infectious mononucleosis, did not have significant pre-illness baseline differences. So stress, coping, anxiety, or depression wasn't significantly different at baseline pre-illness for those who would end up not recovering versus recovering. We did found some baseline pre-illness deficiencies in immune markers of the group that went on to develop severe ME/CFS versus those who recovered following mono. The deficiencies were in production of IL-5, IL-13 prior to contracting IM, and that might influence immune response and immune dysregulation once the virus is contracted. It suggested some predisposing irregularities in immune response. Our next study looked at networks of cytokines, and membership involves groups of cytokines. Modularity involves the validity of the membership categories. Eigencentrality is the importance of the cytokine in connecting a network, and mean degree is the average number of connections a cytokine forms with others. This is kind of what the cytokines look like. On the left, you can see controls. On the right, you can see severe ME/CFS. And basically, the red are pro-inflammatory cytokines. The blue are anti-inflammatory cytokines. The gray are mixed or unknown. You can certainly see very different patterns in these two groups, controls versus those that are severe. Those who developed severe ME/CFS following mono had more centrality or density of connected cytokine networks at baseline, pre-illness. This is 6 weeks prior to at least the onset of IM. They had more differentiated cytokine networks, and they were seen for the recovered controls, and more similar network methods. In a pediatric community-based sample, we also found a similar activation of inflammatory mechanisms. Study 3 involved predictions. We were looking at ROC, receiver operating characteristic, that's the statistics, and we had 2 random forest analyses conducted, separate patients with severe ME/CFS versus controls. One, we just used the DSQ, DePaul Symptom Questionnaire self-report items, and one with cytokines. Findings were the patients with stomach pain, bloating, and symptoms of irritable bowel at pre-illness baseline prior to infection, low levels of IL-13 and IL-5 at pre-illness, and gastrointestinal symptoms when they contracted mono. Those folks who had these characteristics had nearly an 80% chance of developing severe ME/CFS following infectious mononucleosis. Those who developed severe ME/CFS 6 months following IM had more baseline gastrointestinal symptoms and abnormal immune markers, and worse gastrointestinal symptoms at the time they developed IM. It's of interest that the original Fukuda case definition did not include gastrointestinal symptoms among those who qualified for a diagnosis of ME. There's other data sets that also point to some of these gastrointestinal issues. In an adult survey of participants reporting having developed ME/CFS prior to age 18, 40%, or 246 out of 617, had experienced gut problems. And in a factor analysis with 299 patients with long COVID, during the first 2 weeks of gastrointestinal factor emerged, which was not present at 6 months. In terms of the cytokine factors, IL-13 and IL-5 were implicated as risk factors for the development of severe ME/CFS following IM, and there's evidence in human and mouse models that these cytokines contribute to pathology with ulcerative colitis. The 4th study that we conducted was metabolic pathways prior to mono. We examined baseline levels among those with severe ME/CFS versus controls. We used non-parametric significant tests, and we had a significant level of 0.01 divided by 265 because that's how many comparisons we had. So it had to be very significantly different for us to count. The severe ME/CFS and recovered groups were then classified into binary logistic regression with significant features imputed into a series of models with the leave one out cross-validation technique. And then a series of binary logistic regressions were conducted to classify the severe ME/CFS and recovered groups. Significant differences in peak area values between the severe ME/CFS and recovered groups were observed for 8 metabolites. And you can see these significant results at baseline prior to individuals getting infectious mononucleosis. And again, we're looking at those who, in a sense, went on to develop ME/CFS 6 months later versus controls. And you can see that these are the types of findings that we had. And this is a little bit of a description of these particular variables. I don't have time to go into them here. But overall, the results produced, we were able to correctly predict those with severe ME/CFS versus recovery controls with an accuracy of 97%. We were able to identify potentially dysregulated pathways that are essential for proliferating cells, particularly during a pro-inflammatory immune response, and thus consistent with irregularities in cytokines that have been reported. We're now doing a 6 and 7 year follow-up. We've received additional funding. We're comparing the baseline data in behavioral immune from 6 to 7 years ago with the current state. And we're also recruiting patients now with pediatric long COVID to compare with their samples of those with ME/CFS. That's ongoing research. Some of the implications for long COVID with our work for the last decade. we've learned lessons from the ME/CFS case definition that can be instructive for those working with long COVID. There's 3 issues I'd like to briefly mention. One has to decide on which symptoms are contained in the case definition, and that's what the IOM and Canadian criteria did. One needs to develop valid instruments to reliably assess those symptoms, and one type questionnaire that we have been using is the Pulse Symptom Questionnaire. One has to specify thresholds regarding whether or not a particular symptom is severe and frequent enough to meet criteria. The first issue has to do with criterion variance. Criterion variance involves differences in the formal inclusion-exclusion criteria. It accounts for the largest source of diagnostic unreliability. Criterion variance occurs when operationally explicit criteria do not exist for diagnostic categories or when there are varying criteria. When diagnostic categories lack reliability and accuracy, the validity or diagnostic category is limited. And there have been problems of criterion variance certainly have plagued past case definitions. So in other words, you can't just ask patients if they have ME/CFS. Working with ME/CFS, we've learned it's important to use methods that can rely on more than just asking them. In adult and pediatric community-based studies, 90 to 95% of individuals we've identified are not even aware they have ME/CFS. In one of our recent studies with a sample of over 400 people with long COVID, of respondents who reported they had ME/CFS, 29% did not meet criteria based on case definitions. Of those who did report they had ME/CFS, 40% nevertheless did not meet criteria. So both overdiagnosis and underdiagnosis are evident. It indicates that you really need to use a case definition, and you have to decide on what symptoms. There's hundreds of potential symptoms, and one has to make some decisions on that. Second critical task is to use validated questionnaires to assess symptoms. We've been using the Pulse Symptom Questionnaire. It's a 54-item scale. We also have developed a 14-item scale, a short form that can be completed in 5 minutes and can give you a particular case definition, and it's being used in quite a few different countries around the world. Let's talk about fatigue real briefly. Just as an example, fatigue affects 25% of the population, but that's not specific to ask whether a person has the occurrence of fatigue. So we've just learned that occurrence-type questions don't differentiate ME/CFS from other conditions. The same situation probably occurs with long COVID. So what happened was people switched from occurrence to frequency of symptoms, but problems still occur with trying to differentiate, for example, ME/CFS from psychiatric conditions like major depressive disorder. So you can see from ME/CFS and major depressive disorder, MDD, there's no difference if you look for the occurrence of fatigue, and that's part of the problem. So investigators moved to adopting severity measures because of limitations of occurrence and frequency measures. And if you look at the severity of fatigue, before we looked at the frequency of fatigue, this is the severity. Now you can see the ME/CFS is significantly different from the MDD, major depressive disorder, and control. So some symptoms occur at high severity, like migraine, but only infrequently, once every 2 months. So just severity will not capture the burden of a symptom for a patient. So we've been using something called the Pulse Symptom Questionnaire, and you can see that has frequency and severity on it. If you basically make the cutoff 1 or greater, a little of the time or mild, you have difficulties. These are classic symptoms. The light blue is ME/CFS patients. The darker blue is controls. And you can see that controls have a lot of these symptoms at a 1-1. But if you make the classification a 2-2, you can see that you can differentiate the patients from controls very easily. So 3 tasks facing long COVID researchers: to specify symptoms as occurred with ME/CFS, because there's hundreds of possible ones, to develop psychometrically sound instruments with adequate reliability and validity, and to specify thresholds for when a symptom can be considered a burden. And I'm going to examine a couple studies real quickly that have used these characteristics. In one study by the Utah group, Suzanne Vernon, working with the Pulse Symptom Questionnaire short form, patients with ME/CFS and long COVID over a year period of time, at baseline, those with long COVID group had similar symptom scores as patients with ME/CFS. and 5 symptoms improved significantly over the course of 1 year, including fatigue, PEM, brain fog, irritable bowel syndrome, and feeling unsteady, but no significant improvement for ME/CFS patients over time. In a second study by Gargle, examined those with ME/CFS and long COVID. They trained a single class and multiple binary random forest models to assess predictive accuracy, sensitivity, and specificity of the pulse symptom questionnaire short form in discriminating between long COVID and ME/CFS. They ran 30 iterations for each classifier. There was an 80-20 split between training and out-of-bag steps. And it's very interesting that cold limbs and flu-like symptoms were the best discriminators of the two groups. And yet these two symptoms are frequently not on most COVID questionnaires. Third study by Hui built predictive models based on forest algorithm using participant symptoms for initial weeks of COVID-19 infection to predict if participants would go on to later meet criteria for development of ME/CFS. Early symptoms using the pulse symptom questionnaire, particularly those assessed in PEM, predicted the development of ME/CFS with an accuracy of 95%. Finally, I wanted to mention clinical versus research. When classifying patients with broad clinical versus narrow research ME/CFS case definition, you can really make lots of significant differences. I've talked about a number of these studies up till now, but again, the precision of what we're doing increases when we can sort of differentiate patients into these two groups. My final comments: considerable work needs to be done in the long COVID area involving specifying symptoms, developing sound psychometric instruments to measure the symptoms, and establishing thresholds that symptoms need to meet to signal if it's a burden. Insights from ME/CFS research could help in the development of research with long COVID, ultimately improving our efforts to understand its pathophysiology, early diagnosis and prognosis, as well as to identify effective treatments. Thank you.
Thank you, Leonard. Thank you very much. And thank you also for developing this very helpful questionnaires. I think these really made a big change in clinical research by better defining patients. Actually, we use a similar approach now based on your assessment of frequency and severity to quantify symptoms in ME/CFS patients. And this is the basis for our German ME/CFS registry, which was built up by Uta Behrens. So are there questions to the first part of the presentation? Yeah, please.
Thank you for your presentation. I am very happy to see that there are new methodologies and methods being used to analyze data, such as machine learning algorithms and also network analysis. And I saw some results that you have, and you discuss about S-adenosylmethionine, and you mentioned glutathione and cysteine. My question is, what are some other methods that we can use in order to try to better understand where all of this is coming from?
Thank you for that question. I think really the point of my presentation is kind of methodology is so important for our area. I think the ME/CFS field has struggled with trying to identify who has and who does not have this illness. And if different laboratories around the world have different patient populations, it's going to be much more difficult to find consistent results. With long COVID, I think the same issue is going to occur. Even though SARS-CoV-2 is the precipitating event, we're going to have very similar issues of trying to decide at what point do we call a person having recovered, maybe having some minor symptoms, versus those who have major symptoms, and how do we differentiate those who have long COVID versus those who have recovered? You think that's kind of an easy issue. It's actually pretty complicated. And again, I think that if we can make better ways of differentiating these groups into categories that are replicated across different labs, I think the kind of consistency of our results will actually enhance the dialogue that's occurring at scientific quarters.
Okay, so thank you very much. Thanks again joining us, and hopefully looking forward to see you in the next year here in person.
Thank you.